Good morning students, this is your teacher Neelam Elawat and today we'll start our revision with the second chapter, that is sexual reproduction in flowering plants. Dear students, I hope there is no queries in the chapter number 1 and you have thoroughly revised, or you can say you have prepared the chapter number 1 for the test. Isn't it everyone? Okay, so today we are starting with the revision of chapter number 2, right? And I hope you are ready. Your books, notebooks, they are ready? Yes. So let me ask you a question. What do you understand? What is a flower? Any idea what is a flower? Yes. What is a flower, dear? Have you seen a flower? Yes. And isn't they beautiful? They are in different colors. Yes, so when you look at the flower, you find them amazing, isn't it? They are so beautiful, they are of different colors, they are very attractive, isn't it? Right, so flowers are basically the object of aesthetic, ornamental and social and religious and cultural value, isn't it? They are used as symbols for conveying your important feelings such as your love, affection, happiness, isn't it? But flower actually is a modified shoot. Flower is a modified shoot and it is a reproductive structure of angiosperms. Angiosperms means it is that class which contains those plants that bear flowers. And in terms, if you look at the flowers is basically these different parts, right? It has stigma, style and ovary. This is ovary. Stigma, style and ovary, they together constitute style. Okay, that together constitute style. So we can basically categorize the different parts of the flower. One is, can you name anyone? One is sepals. Okay, the very first is sepals. Then is your petals. Okay, then is a stamen. And fourth is the pistil. Now just try to locate all these 4 parts in the picture. Sepals, petals, okay, anther, filament. Sepals, they are generally called as calyx. Okay, sepals, they are collectively called as calyx. Petals, they are called as corolla. Stamen is called androecium. Okay, what is stamen called? It is called as androecium. Let's write it somewhere else so that you can clearly observe. It is called as androecium. And pistil is called gynoecium. Okay, pistil is called gynoecium. That is gynoecium. So flower is basically composed of 4 parts: sepals, petals, stamen and pistil, which are scientifically called as calyx, corolla, androecium and gynoecium. The stamen is a male part of the flower, or you can say androecium is a male part of the flower, gynoecium is a female part of the flower. Okay, androecium has 2 parts, anther and the filament, while gynoecium has 3 parts, stigma, style and ovary. So if we define flowers, so flowers are morphological and embryological. Okay, they are morphological and embryological marvels and are the sites of sexual reproduction. Okay, they are the sites of sexual reproduction. Now coming to the pre-fertilization event. Much before the actual flower is seen on the plant, right everyone? There are certain processes, or you can say the plant has certain decisions to take before the flower formation takes place. And the cultivation of flowering plants is called floriculture. Okay, this is a very important term. So what is floriculture? Cultivation of flowering plants. And when we are talking about, there are certain pre-fertilization events that will be occurring in the plant. Several hormonal and structural changes are started, which lead to the differentiation and development of the floral primordium. okay, that are lead to the formation of a floral primordium. Then what is basically a primordium? Primordium is an organ which is in earlier stages of development, lays the foundation for the development of the flowers. Inflorescences are formed which bear the floral buds and then the flowers. तो एक तरीके से inflorescence बनती है जिससे बाद में flowers की formation होती है. And as I said, flower has two main parts, androecium and gynoecium. Androecium is your male part and gynoecium is your female part, and both these structures, they represent the male reproductive structure and the female reproductive structure. So as I said, androecium has two main parts, okay, or androecium is commonly called as stamen. So stamen has two parts, one is the filament, another is the anther. Now just look at the diagram. This long part is called the filament and this portion is called the anther, right? So stamen is made up of two parts, anther and filament, okay? It is made up of two parts, anther and filament. Okay everyone? Right. If there is a single theca, okay, if it has single theca, then we call it as monothecous, and if it is you can say if it bears two thecas, then it is called dithecous. For example, is your brinjals. Your brinjal is dithecous while hibiscus is monothecous. The proximal end of the filament is attached to the thalamus. Okay everyone, the proximal end of the filament, this portion is attached to the thalamus. Okay, this will be attached to a thalamus. The number and the length of the stamen, it varies. Okay, the length of this filament and the number of filaments, how many filaments will be there, that varies, and this filament is attached with the thalamus. Okay, then coming to the anther. Anther is dithecous. The anther is bilobed. Okay, it has two lobes. See, there are two parts. If you look at this is part 1, part 2. So anther has two lobes, so we call it as bilobed. So, and it has two theca. Each lobe has two theca. See, there are 4 parts. You can see in the picture there are 4 parts. See, there are 4 parts, isn't it? So there are two lobes which are present and each lobe has 2 theca. So there are 4 thecas in total. Okay, there are 4 thecas in total. Same if we cut it, we observe the same part. Here also we can see 4 thecas and this is 2 lobes you can easily locate. So your anther is bilobed and each lobe has 2 thecas. So we call it as, so in total it has 4 thecas. Now just look at this, the diagram. It is very, very clear. So when we cut it, the transverse section, when we cut it, okay, with 3-dimensionally we cut the section of the anther, then we observe that there are 4 thecas. Clearly we can see 4 thecas, isn't it? So there are 2 lobes and each lobe has 2 thecas. So total there are 4 thecas. Okay, clear everyone? Now see this, just look at this picture you observe. Just observe this picture everyone. Okay, have you observed? Now just look at this. We know that the anther has a 4-sided structure, isn't it? The anther has 4-sided structure. See, you can see the 4-sided structure, isn't it? So there are 4 microsporangia which are present, right? There are 4 microsporangia which are present in the corners. Can you see the brown structures? They are your microsporangias. Okay, so there are 4 microsporangias. So 2 are present in each lobe. Okay, and these microsporangia, they will develop further and will become the polar sac. ठीक है? So हमारे पास 4-sided structure है जिसके अंदर 4 microsporangia मौजूद है. Clear? और इन microsporangia से बाद में क्या बनेगा? Pollen sac will be formed. Okay everyone? So pollen sacs will be formed. Clear? Is it clear to everyone? Yes. Now let's look at the structure of a single microsporangium. Let's understand the single structure. Just look at this. The microsporangium, it is surrounded by 4 walls. Okay, 4 walls, it is surrounded with 4 walls. The outermost wall is epidermis. The outermost is epidermis. Right, everyone? And the second is endothecium. Okay, second is endothecium. Okay, second layer is endothecium. Outermost is epidermis, second is endothecium, and the third is middle layer. Okay, and the third is middle layer, and then we have a tapetum. Right, then we have the tapetum. The outermost 3 walls, we should label it, isn't it? There is epidermis, then you will have endothecium. Let's label it endothecium. It is endothecium. Then this one is middle layer, and then you have tapetum. The outermost 3 layers, okay, the outermost 3 layers, they are protective in function. Okay, they are protective in function, and they keep the anther moist. Okay, they keep the anther moist, and the innermost layer is called tapetum. Now this tapetum, it provides nourishment to the pollen grains. See, inside there are pollen grains, isn't it? Are they visible to you? So inside it there are pollen grains are present. So tapetum, it provides nourishment to the growing pollen grains. Okay, clear, everyone? So your microsporangium, it is surrounded by 4 layers: epidermis, endothecium, middle layer, and then the fourth is tapetum. Outermost 3 layers, they provide protection, and they keep the anther moist so that it can easily release the pollen. The innermost layer, that is tapetum, it provides nourishment to the growing pollen grains which are present inside it. Okay, everyone? So your pollen mother cells, they are diploid. Okay, the pollen mother cells, they are diploid, right? Now this pollen mother cells which is diploid, it will undergo the meiosis process. It will undergo the meiosis process to form tetrad. Okay, to form the tetrad. Okay, 3 are visible, 1 is on the back side. So there is formation of 4 pollen grains. Okay, so in the pollen mother cell, the pollen mother cell will undergo the meiosis process, and after meiosis it will form tetrad condition will be there, and after that there will be formation of 4 pollen grains. Okay, everyone, there is a formation of 4 pollen grains, and this process where 4 pollen grains are formed from the pollen mother cell. Okay, this process where 4 pollen grains, they are formed from the pollen mother cell, right? From the pollen mother cells, that is your PMC. Okay, that is your PMC. We can write it as PMC, polar mother cell. So this process is called microsporogenesis. What do we call it, everyone? Microsporogenesis. What do we call it as? Microporogenesis. Okay, this is called microporogenesis. Right? You can again see, again I am showing the different picture. You can have again the different layers, that is I was talking about: epidermis, endothecium, middle layer, and the microspore mother cells are present. Then they are being nourished by the tapetum. Tapetum will provide nourishment to the growing pollens, and this microspore mother cell, it will undergo the meiosis process, and it will form 4 microspores. It will form 4 microspores or 4 pollen grains, and this process is called microsporogenesis. Okay, what do we call it as? Microsporogenesis. Clear, everyone? This is called microsporogenesis. Clear? Now these 4 microspores, okay, they will later form the 4 pollen grains. ठीक है, जो 4 हमारे पास microspores बने हैं. So you have PMC. From PMC you will have 4 microspores. Okay, and these 4 microspores, they will form pollen grains. So you have formation of 4 pollen grains. So microspores, they will form pollen grains. Okay everyone? Microspores, they will form 4 pollen grains, and this process by which the microspores are formed is called microsporogenesis. Okay everyone, this is called microsporogenesis. Now let's understand the next topic, that is, so let's briefly again decide with, so your diathecus, right, your anther is diathecus, it is bilobed. Anther is surrounded from, it is a 4-sided structure having 4 microsporangia which are present in the corners. Then microsporangia, they will develop to form the pollen sacs, isn't it? They will develop to form the pollen sacs. Then there are 4 layers again: epidermis, endothecium, middle layer, and the tapetum. Tapetum will provide nourishment to the growing, you can say growing PMC. Then what will happen? Then PMC will divide, undergo meiosis, and it will form 4 microspores. This process will be called as microsporogenesis, and these microspores will later form pollen grains. So you can see, can you see the pollen grains? Is it visible to you in the diagram? See, so much amount of pollen grains are being formed. So when the anther is young, a group of compactly arranged homogeneous tissue is there, which is called a sporogenous tissue. Right? Sporogenous tissue is present in the center. Right? It is present in the center, and the cells of sporogenous tissue will undergo the meiotic division and will form the microspore tetrad. And this process we call it as microsporogenesis. What do we call it as? Microsporogenesis. So let's come to this. Just understand, we have microspore mother cell, that is PMC. Okay. So we have sporogenous tissue. Sporogenous tissue will lead to the formation of PMC. PMC will undergo the meiosis process. Okay, and it will form microspore tetrad, and this process is microsporogenesis. So there are 4 haploid cells are formed, and they will stay together. All these 4 cells, they will be together. Then when these microspore tetrads they mature, they form the pollen grains, and then the tetrad will separate. Okay, the tetrad will separate. So 4 pollen grains, they will be released. Coming to the structure of pollen grains. The pollen grains, they are referred to as the male gametophytes. Okay, the pollen grains are referred to as male gametophytes. Okay, let's discuss the pollen grain. So pollen grain represents the male gametophyte. Okay, it is of various size, various shapes, colors, designs. So different species have different kinds of pollen grains. Some of the pollen grains, they are well preserved as fossils because of the presence of sporopollenin. Okay everyone? Clear? So your pollen grains, they can be well preserved as fossils as they are covered with sporopollenin, and it's a very hard covering. Clear? It's a very hard covering. It is this material can stand high temperatures and even strong acids in alkali, and it is the most resistant organic material which is known today. Clear? They can also be cryopreserved. The pollen grains, they can be cryopreserved. That is, they can be kept at a temperature of -196 degrees Celsius and then can be used for crop breeding. Pollen grains, they have a nutritional value. Okay, that is why they help in increasing the performance of athletes and race horses. But in some cases, pollen energy has also been observed in few people, right? Like people who have asthma, bronchial infections. So in that case, it also causes the allergies in certain people. The viability of the pollen grain, it depends upon the temperature and humidity. Means once the pollen grains, they are released, then for how much period they are viable, they are able to fuse with the, or they are functional, it depends on the temperature and the humidity. Some species have the pollen grains which are viable for few minutes, and some they are for several months. Right everyone? Okay. So examples we can quote here is Solanaceae, Rosaceae and Leguminosae. These families we can quote as an example. Now pollen grains, they are covered with two layers. The outermost layer is called the exine, and it is the outermost is made exine is very hard because it is made of sporopollen, and the internal layer is called intine. So you can very easily designate here. See, this is one of the pollen grains. Exine is labeled here. Now look at this. It has both of them. So outermost layer is called the exine, and the inner layer is called the intine. So a pollen grain, it is surrounded with two layers. Outer is exine, internal is intine. Exine is made up of sporopollenin. Okay, and exine has certain apertures which are called germ pores. तो जो हमारी exine layer है वो sporopollenin की बनी है, और कुछ स्पेसेस में germ pores मौजूद हैं, और जहाँ पर germ pores मौजूद है वहाँ sporopollenin is absent. Okay, sporopollenin is absent. The innermost layer is called the intine, and this layer is continuous. Okay, it is very thin and it is continuous, and this layer is made up of cellulose and pectin. So exine is made up of sporopollenin and intine is made up of cellulose and pectin. Clear? Okay, let's understand. As I said, the pollen grains, in certain cases they cause allergies. So if you inhale them, if they are inhaled, then in that case they will cause allergies. Clear? Now coming to the next that we have to talk about, let's look at the structure of the pollen grain. Let's start it from here. The cytoplasm of the pollen grain, it is surrounded by a plasma membrane. So outermost layer is called a plasma membrane. Okay, and then inside it has exine and intine. So outermost layer जो है वो कौन सी है? Plasma membrane. फिर आपकी exine है, and then it is intine. Clear? During development, जब microspores divide होते हैं, microspores, you remember tetrad is formed, meiotic condition. So वहाँ पर हमारे पास 4 pollen grains की formation होती है. 4 pollen grains are formed, right? Now this pollen grain microspore, it undergoes a mitosis and it forms 2 cells. Just look into the diagram. It forms 2 cells. The first cell which is bigger in size, it is called the vegetative cell. The cell which is smaller in size, it's called the generative cell. Okay, vegetative cell which is bigger in size and it stores food, and it has a irregular shaped nucleus. So this is your vegetative cell which is bigger, and it has this pink color is your nucleus. Generative cell is smaller and it also contains a nucleus. Now 60% of angiosperms, in 60% of angiosperms, the pollen grain, they are shed at this 2 cell stage. जो हमने अभी भी बात की है, 2 सेल स्टेज होते हैं. एक बड़ा सेल होता है जिसको हम जनरेटिव कहते हैं, एक छोटा सेल होता है जिसको हम एक बड़ा सेल होता है जिसको हम वेजिटेटिव सेल कहते हैं, एक बड़ा सेल होता है जिसे हम जनरेटिव सेल कहते हैं. वेजिटेटिव सेल के अंदर एक स्पिंडल शेप्ड न्यूक्लियस है, एक बड़ा सा इरेगुलर शेप न्यूक्लियस है, लार्ज न्यूक्लियस है. जनरेटिव सेल के अंदर स्पिंडल शेप्ड न्यूक्लियस है, राइट. सो 60% पोलन ग्रेन्स में इस स्टेज में पोलन ग्रेन इज शेड, while in some other the pollen grain, or you can say one that is 2 male gametes, okay, the pollen grain, it is shed at 3 cell stage. Okay, in 60% the pollen grains, they are shed at 2 cell stage, while in 40% of the cases the generative cell, okay, which cell? The generative cell will divide. The smaller cell, which is your generative cell, will divide and it will form 3 cell stages, and they are shed in 3 cell stage. For example, your jawar. So jawar is, in case of jawar, the male gametes, they are shed at, or you can say the pollen grains, they are shed at 3 cell stage. Now coming to your homework, dear students, first of all you have to revise chapter number 2. And you have to complete question answers 1 to 5. You got to thoroughly revise this portion of the, you can say, chapter that we have talked about the microsporangium, formation of microsporangium or the stamen. You will thoroughly revise it and you will learn the question answers 1 to 5. Okay, everyone? So that's all for today. Bye, everyone.